Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton, and today guys we are going to be continuing yet again with our gene technology section of A22 biology, and we're going to be talking about gene therapy this time. So you have GMOs, gene transfer, genetic fingerprinting, PCR, gene therapy, and I think there might be one or two more as well that we have to go through. But once we've done that there, it should be pretty much straightforward. We'll just have the plant kingdom, the animal kingdom, we'll have dihybrid crosses, monohybrid crosses, selection, speciation, a couple of more videos to go, like I said, but right now we'll talk about gene therapy. So what is gene therapy? Well, gene therapy is the treatment of such diseases by introducing a functional allele of the gene into the affected cell. So cystic fibrosis and muscular dystrophy are caused by the presence of a single defective gene, and these defective alleles are recessive. So a functional allele then is inserted into the cell which is affected by these conditions. There's two distinct approaches you can use. The first is somatic cell gene therapy, which will only target the affected tissues where the condition is caused by a defective gene, and this will affect a specifically easily reached part of the body, and this can be used at any stage of your life. Then you have germline therapy, which is whenever you replace the defective gene in a fertilized egg, and all cells will develop the individual normal allele with no issue targeting the affected tissue. The defective gene will not pass on to the future offspring, and gametes will not contain the defective gene. So, inserting then, how do we get this functional allele into recipient cells? Well, you can use adenoviruses which cause respiratory infections, as you can inject DNA into lung epithelial cells which have a harmful disease-causing gene being removed, and the donor DNA will be spliced in. It is difficult to remove disease-causing genes without dismantling genes allowing a virus to enter the cell, which will cause infection, and some individuals may be immune to adenoviruses, and therefore they'll be unable to penetrate these cells. You can use retroviruses, which is when the RNA will be converted by reverse transcriptase into the DNA in the host cell, so the donor DNA will replace the defective DNA in the host cell. You can use liposomes, which is artificial lipid vesicles, which will contain the donor DNA. The lipid coating proteins against degradation will help DNA bind to the target cells. The problems we now know about gene therapy is that DNA is incorporated into the host DNA, or independent DNA units are not incorporated into the main nuclear DNA. Difficulties at targeting all affected areas is treatment is usually short-lived, new and replacement cells don't have the DNA, and offspring of treated individuals are still affected. Genetic disorders also may involve many genes across a range of chromosomes, so it's actually quite difficult to identify the genes that are involved and effectively incorporate all the DNA necessary into a host cell. Cystic fibrosis is a genetic disorder that results from a mutation of a gene that causes for a carrier protein called CFTR, otherwise known as cystic fibrosis transmembrane conductance regulators. And this is whenever and how we do this is protein transports chloride ions out of cells, and water will fall by osmosis. So CFTR is not working, and therefore there's less water and more mucus is produced, and therefore it's thick and sticky, and that's how cystic fibrosis actually occurs. The problems you get with this mucus buildup is that the thick mucus will collect in the lung, and this will interfere with gas exchange, increasing the chance of a bacterial infection. The pancreatic duct will also be blocked, and this will interfere with digestion in the small intestine, and reproductive passages can be blocked, making a person sterile too. The treatment using gene therapy for cystic fibrosis is that you can insert the normal allele for cystic fibrosis, so it doesn't code for cystic fibrosis, into the modified adenovirus, and you can allow this virus to infect the cells in the respiratory passages, introducing the gene, which therefore means cystic fibrosis will not occur. You can also insert the normal allele into a liposome and spray these as an aerosol into the person's respiratory passages. Problems of using this treatment though is a few cells will actually take up the normal allele, cells will not live for long, and the treatment needs to be repeated every few weeks. By using adenoviruses, people may develop serious lung infections, and it's only possible in cells where the respiratory will take up the normal allele and not cells in the pancreas or reproductive organs. Thank you for watching this video. I've been Dylan Hamilton from C.A.R. Entertainment, and I will see you next time.